It is now my great pleasure to introduce our third Nobel laureate, Dr. Fred Ramsdell. Dr. Ramsdell was born in Elmhurst, Illinois. He earned his Bachelor's of Science degree in Biochemistry and Cell Biology from the University of California, San Diego. He then moved up the coast and did his PhD at the University of California, Los Angeles, and graduated from there in 1987. Following this, he transitioned to the NIH, where he performed his postdoctoral training, and he focused there on immunology and specifically immune tolerance. He then had a stint of working at a number of companies, first at Immunex, and in 1993 he joined Darwin Molecular Corporation around the same time as Dr. Mary Brunkow, as you just heard. At Darwin, he was the person who established the immunology program, and which then led to the groundbreaking work that we're about to come. Between 2004 and 2008, Dr. Ramsdell joined and worked in many companies, including Zyme Genetics. He established the Inflammation Research Center in Seattle, which was part of the Novo Nordisk Seattle site. He also worked for a while at Atar Pharma, and then he was a chief scientific officer at the Parker Institute for Cancer Immunotherapy in San Francisco. Following this, he co-founded Sonoma Biotherapeutics, which is a company that now works and focused on the development of regulatory T cells as therapies. And this company was co-founded with Jeffrey Bluestone, Xixing Tang, and Alexander Rudensky. And he was the chief scientific officer there until just recently. So for our last speaker this afternoon, it's my great pleasure to introduce Dr. Fred Ramsdell to the stage to give his Nobel lecture.
Thank you very much for the kind introduction, and I want to thank the Nobel Foundation and particularly the Karolinska Institute, the staff and faculty here who have been absolutely fantastic. And I think all three of us as laureates consider this to be an amazing award, and it's a very humbling experience. But I think we're learning to enjoy it finally. I'll throw some disclosures up. That's pretty quick. I'm going to divert and not talk about this for a minute because this really has been a very amazing week, and it's only 2 days old. But nonetheless, we were at the Nobel Museum for the last couple of days, and you may or may not know that they asked designers to design a dress for each one of the disciplines every year. And this is the dress for physiology and medicine this year. And the dress is called U, Y-O-U. And it's designed by two young women from Beckman's design group, Odessa Galloway and Lovisa Schmidt, who I did not get to meet, but I would love to one day, because when I looked at this from the back, I immediately saw this interesting pattern, which only a T-cell dork like myself would really notice, which is that if you look at the part of the dress here that kind of comes up and it's got a long train, and this is the Y part of the U. Okay, there's a little bit of this is a mannequin, so there's a little bit of midriff there, and then there's this circle and this other shape above that circle. And so I looked at that and said, this looks exactly like a T-cell receptor binding to an MHC molecule that has a peptide in its groove. And the very lovely woman at the Nobel Museum said, that's exactly right. And I thought, how in the world do you know that? That's incredible. And more importantly, how do these two people know that? That's incredible. I still don't know the answer to that, but I'd love to find out. But if you looked at the diagrams on the back, that was clearly exactly what they were trying to do. So I was incredibly impressed. So even the dress designers in Stockholm apparently know an awful lot about immunology. All right, so we'll come back to why I'm actually here. It's not to talk about dresses, which is the scurfy mouse and how that's led. And I'm going to tell a little bit of a story and bring us kind of full circle into where we are literally, at the moment in terms of how we're trying to advance these programs. So this is a timeline that was put together by my friend and former boss, Jeff Bluestone, Megan Levings, and Victoria Fung at UBC. And Whoa, I gotta go back again. Sorry, Mary. Okay, Mary, got it. All right, so we're gonna, dang it. You know, I knew I was going to do this. All right, so we're going to go back to the early 40s and 50s and 60s. And in 1960, there was an award to McFarland Burnett and, oh my God, how am I blanking? Whoever else is listed up there. For the concept of peripheral tolerance. That happens to be the year I was born. So 65 years ago, the Nobel Committee awarded this Nobel Prize for the concept of peripheral tolerance and the ability of people to control responses to their own tissues. So we're going to eventually fast forward 65 years to today, where another Nobel Award it recognizes the same thing, which is pretty amazing. It's either amazingly quick or amazingly slow, depending on how impatient you are. I consider it slow because I'm pretty impatient. But I'll just tell you why we were working on this again and remind you of this. And this is data from Lisa Clark, who was in the lab at the time. And when we got the scurfy mice from Oak Ridge National Labs, and Mary has talked about this, as has Shimon previously, this is just some of the data that we generated. These mice, in many, many different ways, were hyperactive. If you took the lymphocytes out of these animals, the T cells out of these animals, they responded incredibly quickly. Normal cells need two signals. So this is shown over here. Normal cells need two signals to give you a good response. This is looking at those cells' ability to divide. This is to make cytokines. It's basically the same answer. Scurfy cells don't need that second signal. They divide like crazy, and they don't seem to care, which was fascinating to us. So when we generated the transgenic mice, and Mary has told you all about the cloning of this gene and the fact that it was the last gene in the list of possibilities, I'll come back to that, was a little distressing to us at the time, or at least certain to me. But when we cloned the gene, we also made these transgenic mice that Mary talked about. So this is another example of again, stimulating these lymphocytes out of these animals, and this is more stimulation, less stimulation, more response, no response. Normal animals look like this. The mutant animals, again, hyperactive, but these transgenic animals were hypoactive in every assay we looked at. And as she said, we had 5 different lines of mice. The more of the scurfin protein that was present, the less responsive these cells were. So it was the yin and the yang of responsiveness, and it was I called it a rheostat for functionality at this particular point in time. And we didn't know what that meant, but we thought that was a really good hook into how we might use these cells. And we also were aware of the really pioneering work from Shimon and his colleagues published here, but we always cite this one paper, but there's more than one paper. There's a ton of work looking at this small population of cells in the mouse that had CD25 and CD4, and Shimon's described these quite well. Roly Kotri in the lab did this experiment very early on and showed that if you isolated just these cells, those are the only cells in the hematopoietic system within the T cell lineage or any other lineage that express the FOXP3 gene. So that, to me, was a revelation. So that obviously provided a molecular marker for what these cells were, but it also meant that this gene, at least it meant to me, this gene drove these cells and was really responsible for that particular lineage. It was also responsible for their functionality. So this is another way of showing some data that Shimon's already shown in principle, which is when you put normal T cells into a system, you can stimulate them and they'll give you a response. If you add regulatory T cells, so these Shimon Treg cells, it will inhibit that. If you add non-regulatory T cells, you get an increased response. We knew that. That was perfectly predictable. When we looked at the transgenic cells, however, again, the regulatory cells from these transgenic mice were very good at inhibiting, but now these cells which don't normally have suppressive function now do. So again, FOXP3 expression in any T cell can drive at least some aspects of a regulatory program and a suppressive program, which was amazing to us. So we also knew that these cells are derived from the thymus, and we asked, well, what happens if we only express this gene within the thymus? And so when the cells then leave the thymus and go out to the periphery, what will happen in that case? So we did that experiment, and it turns out expression in the thymus gives you thymic Tregs, but it does not prevent disease when you cross these animals, that gene onto scurfy mice. So this, and forgive the nomenclature, but this is the transgenic mouse you've seen before. So if it has the transgene, the normal FOXP3 gene, you don't get any disease. But if it has that same gene, same FOXP3 gene that's only expressed in the thymus, these animals still come down with disease. It's the same way scurfy does. So expression in the thymus and programming and driving the initial differentiation of these cells is not sufficient. You need to have this gene on in the periphery of these animals. This is shown in a very elegant, though complicated experiment by a good friend and colleague, Sasha Radinsky, much later, but he was able to turn on and turn off the FOXP3 gene genetically within these mice. And what he could do was allow the animals to develop without FOXP3, which means they developed the scurfy-like phenotype. But if he turned FOXP3 on in Tregs at 4 weeks of age, those animals then reverted essentially to a normal phenotype. You can see here they're not totally normal at this particular point in time, but inflammation goes down dramatically and those mice will continue to live. So this really is a dominant form of tolerance in the periphery that can control an ongoing immune response, which is what we need to do in the context of disease therapy. So we were pretty confident at this point that FOXP3 was the master regulator for T-cell development. We certainly had that data. Shimon had that data. And quite quickly, many other people in the world reproduced basically the same essential findings, which was great. It's a necessary gene, and the absence results in a hyperactive immune response, which ultimately results in lethality, whether it's in a mouse or a person, without dramatic intervention. So that was great, and we thought this is a really, really good concept for how we can control the immune system. But, as Mary pointed out, this is a terrible target for therapeutic development. In the year 2000, there was no way to target a transcription factor that made any sense. Even though we tried, we failed miserably at it. And there's no way we're going to actually turn this into a drug. We certainly thought about lots of ways we could either enhance regulatory T-cell number or function, and some of these are shown here. Some of them work to a first approximation, but really they're not therapeutically viable for the most part at the moment. There's some caveats to that, and so there's ongoing work still, but in general, this wasn't really a very fruitful approach back in the year 2000, 2005. We also knew on the tumor side, and Shimon talked about this, that if we could eliminate or at least inactivate these cells in the context of a tumor, we could potentially drive good tumor responses. And certainly that's been shown in the mouse. We think that will happen in the human, and there's ongoing clinical studies, which I won't talk about right now, to try to do that in human tumor settings as well. And we'll see where that ends up in a few years. So I'll come back to this. But because we couldn't target this in a conventional way from a drug perspective, we essentially said, okay, we need to finally get this published and get this out there, and everyone else can work on this and hopefully help us figure this out, which is what we did. And lots of people have helped us out over the years. And in the intervening decade, we learned a lot about how these cells work. There's multiple ways in which they can control an immune response. They can suck up the growth factors from the environment. They can modify the metabolic environment around them. They can deactivate antigen-presenting cells by literally sucking proteins off the membranes of those cells. There's a number of other ways that these cells can control immune responses, and that is almost certainly context dependent on the disease and the tissue, et cetera. We've also learned that these cells are resident through every tissue in our body. In fact, one of the reasons it was the last gene we prioritized to sequence was because the data we had suggested that this gene, this FKH gene, as Mary called it, was expressed in every tissue in our body at a very low level, and I thought that was stupid. That can't be what's causing this disease, so that can't be it. Of course, Completely wrong. These cells are resident in every tissue in our body, and they're really important for maintaining normal homeostasis of our tissues. So these cells not only suppress immune responses, which they're very effective at, and they home very well to inflamed sites, but they also have tissue repair capacity. They will help regenerate tissue. They will help repair damage from an inflammatory response, et cetera. So they're somewhat like having both the police at the site of an accident or at the site of an event, stopping whatever bad is going on, as well as having the medics there who are stopping the bleeding and keeping people alive. So they do a little bit of both functions. And so we've learned a lot about these cells over the years. We've actually learned how to identify them and actually get a good handle on them in the human. You know, all the data you've seen so far is using the CD25 marker in the mouse, which is very clean, works great. Doesn't work at all in the human because we're more complicated than mice, surprisingly. So we did find ways to isolate those. Jeff Bluestone and Kizhi Tong and others at the UCSF identified ways to actually isolate human regulatory T cells. And that happened in the late 2000s, and that enabled some very early studies in which those isolated cells were put into the lab, grown up, and given back to patients. And when that happened, patients were fine, they were safe, they migrated to sites of inflammation. You could see biomarkers of activity as though these cells were doing something, but there wasn't really a meaningful clinical benefit for most of those patients. And so, okay, the simple version of more will fix the problem may not be the correct answer. Again, there's some caveats around that, but in general, it looked like more isn't going to be enough. We're going to have to do something else to these cells in these patients. And so in 2019, right before the pandemic came along, Jeff was convincing in that he could start a biotech company, and Tong and Sasha and myself basically supported Jeff, and I joined Jeff in the company, and we started Sonoma Biotherapeutics. And we really did this because we liked taking advantage of the fact that this is nature's way of controlling immune responses. These are evolutionarily defined cells that have been created over thousands of years of evolution, and their job is to control autoimmunity. So essentially, I don't have to be smart. I just have to use the right cell. And that works perfectly for me in terms of my ability to predict things. So we're using the right cell, we believe. They'll block inflammation. They can inhibit ongoing inflammation, and they can repair tissue, all in one drug. So for us, having this multifunctionality within one drug made a lot of sense, as opposed to some of the other therapeutic approaches that we're doing. So, and by the way, there's numerous companies and other organizations that are actually doing very similar things. I'm going to talk about Sonoma because it's the one I know the best, but there's many other groups that are doing the same sorts of things with their own little twist in different areas. And we can talk about those later if you want at the reception afterwards. So we started with rheumatoid arthritis. There's a number of reasons for that. One, there's great preclinical data to suggest that regulatory T cells can reverse as well as block rheumatoid arthritis in mice. Now, the mouse models are far from perfect, but at least that's a good start. We could show that in arthritis patients, the regulatory T cells don't look as good as they do in healthy volunteers. And there's a number of ways we can show this. It's not that they're absent, they're clearly there, but they're not as active as they are in several assays in human patients. And in fact, the worse you are in an arthritis score, so which is to the right here in this graph, the less good your regulatory T cells look to some extent. Okay, so we thought, all right, they're slightly defective. Can we use those cells, take them into the lab, and make them better and put them back in? Because we think those cells can be resurrected, and there's a whole bunch of data suggesting that would be true, and then we could treat patients with them. We can expand these cells in the lab. This is normal. This is RA patients, again, not quite as well, but certainly good. And the cells look like very good regulatory T cells when we get done expanding these cells. So that's terrific. But what are we going to do to make these cells better? Why are they going to work in arthritis patients? And so we decided we would actually give these cells specificity. So normally, regulatory T cells have a very diverse repertoire of T cells, and some are specific for whatever it is you're interested in. But we wanted to make a lot of ours specific for something that was found in the rheumatoid joint. And so what we chose to make our cells specific to are citrullonated proteins. And so without getting into the details, these are modified proteins, and they're modified during the process of inflammation. And so in an inflamed joint, there are a lot of these proteins that are generated, and they sit in the synovial fluid and in the synovial lining of an inflamed joint. And it's well known, and it's well known largely from work actually from the Karolinska. And this is just an example of showing that there are a lot of these citrullonated proteins within an inflamed joint in an arthritis patient. So we decided we would make a CAR. So this is a chimeric antigen receptor. We would give specificity to our Treg cells. And this is essentially stealing an idea from our oncology colleagues. Clearly, this is a thing that works to treat a variety of, right now, liquid tumors. It's very efficient, but the technology to generate these is actually fairly robust. And so the way you make these is you take a part of an antibody, and you stick it on a part of a signaling domain, and then you stick it, in this case, in a regulatory T cell. Normally, you put these in other cells to kill tumors. In our case, we're just going to focus this regulatory T cell, in this case, on citrullonated proteins. And we did this as a collaboration with Lars Klarsgood and Vivian Mellstrom here at the Karolinska, because really, they are the experts in this. And they have hundreds of these antibodies that were derived from patients. And we selected one that saw a variety of these different proteins with good affinity, good activity, good specificity, et cetera. So the binding part, the business end of our CAR, essentially comes from the Karolinska Institute. Okay, and what you can show is that that CAR Treg now sees things like synovial fluid. It certainly sees purified citrullonated proteins, these modified proteins, sees them quite well, a variety of them, not a problem. It responds to them and does everything a regulatory T cell is supposed to do. But it also sees synovial fluid coming out of RA patients, which was really important to us. We wanted to know that these modified proteins that are found in disease, in the tissue, was going to be able to stimulate our drug product, and in fact, it does. So this drug product, again, it's a regulatory T cell, so it's the whole cell. They all express FOXP3 after we've grown them up, and I'll show you the protocol for that in a second. So they all look like good regulatory T cells by the fact that they express FOXP3. There's other markers as well. This is a little gene expression analysis. I'm not going to walk through this, but the first two columns, in fact, are our drug product. The two middle columns are, in fact, a non-CAR-bearing version of the same thing. And all these red ones on the right are effector T cells. So this is the opposite side of the equation. And so you can see that our regulatory T cells are completely different from these activated, potentially pathogenic cells. So that's not what we're making when we make our product. We did design a manufacturing process for this. And so what we do is we take blood out of a patient. We isolate the regulatory T cells out of that blood, which is a very long process, takes us essentially all day. We put those cells into culture, and we transduce them with a virus that puts the CAR into those cells, and we grow them up for 14 days. And we get billions of cells at the end of those 14 days. They're frozen, quality control checks are done on them, and then they're reinfused back into the patients. And I'll show you some clinical data on this in just a minute. It's a long process, and it's an interesting, challenging process, but it is one that you actually can do. And again, our oncology colleagues have shown it works in that setting. It's not really fundamentally that different here, and so we're working on the backs of the oncologists. And this really just shows that the cells that we get are in fact, if anything, more Treg-like. So this is at day 14 after they've been grown up, and this is a bunch of markers for Tregness. This first one is FOXP3. They're, if anything, more Treg-like after the 14 days than they were going into the culture. So we feel pretty good that these cells are stable and that they're effective, and they will be effective when they get into patients. So this is the clinical trial that's ongoing right now. It's a phase 1 trial, so it's a safety trial. And I can say that so far, every patient who's been dosed is fine. There are no significant adverse events yet. Now, we've done 2 dose levels, so we've done low and middle, and it's only 3 patients each, so it's 6 patients total. So I certainly don't want to overstate the significance of this, but I'll show you the data in a second, and I think this is how we're trying to prove that these regulatory T cells will, in fact, modify disease in a patient population. These are the patients. And one of the things that's interesting I'll highlight down here on these patients is that every patient, so these are the 6 patients I'll show you the data on, every one of these patients has had at least 4 different types of therapeutic intervention. So they've had JAK inhibitors, they've had anti-TNFs, they've had other things, at least 4 different ones, none of which have been effective for these patients. And in some cases, this patient's had 12 different drugs. They've had disease for 28 years. They've gone through 12 different drugs. Their disease continues to progress. So these are patients who, in many cases, are very refractory to the existing therapies that we have out there. So it's a pretty high bar at this particular point to study these patients. So this is the meat slide, if you will, which is on the left here is the axis of it's either tender joints or swollen joints, depending on which one you're looking at. It's either 60 egg joints or 20 egg joints, doesn't really matter in this particular case. These yellowish things are the low-dose group of patients. The bluish things are the mid-dose group of patients. And what you can see in most of them is after the infusion of the product, there's a diminution in the score. So the patients get better. Not all of them, and clearly to varying levels. In some cases, the scores come back up over time. This is 24 weeks. In some cases, the scores come back up. In some cases, they come up a lot. In some cases, they stay down for 24 weeks. In some cases, it's in between. So very early days, but we're very encouraged by the fact that at least we're seeing some response in these patients. There's a lot of other measures that we're taking that I think are even sort of anecdotally useful. Patients can now open a jar, which they couldn't do previous 5 years. We've had patients who have gone for a run, which they haven't done in the previous 5 years. Again, that's all anecdotal. It's small, but I find it encouraging as well. When we look at as much as we can about mechanistic data and what's going on in these patients, this is one patient where we've done some imaging, some ultrasounds. And the top is 2 different joints, and you can see the red, a lot of inflammation. This is 4 weeks after infusion, a little bit of inflammation left, very little in this particular joint. So we can see inflammation dropping by a quantitative measure in this particular case. In some, but not all patients, we're getting biopsies. So this is the medium dose set of patients, and we've gotten a number of synovial biopsies from these patients. One before we gave the infusion, so pre, and 4 weeks after we gave the infusion, post. And this is a score of inflammation. This is the percent of inflammatory cells compared to all cells. And you can see that there's a reduction. In this particular case, a very dramatic reduction in the inflammatory score. 9 is the highest possible value on this particular scale. So it went from 9 to 1 or 2 in this particular scale. So very dramatic reduction in that particular patient. More modest than some of the other patients. But at least we're seeing a reduction in the inflammatory scores, which again, we find encouraging, certainly not proof yet, but enough to keep us moving forward in this kind of trial. We can also, in again, a small number of patients where we have the biopsies, you can't see this, I'm sure, but what these red arrows point to are our cells, so we can specifically look for the cells that express that CAR in this joint. We can find those cells. When we look at the characteristics of those cells, they look like good, regular Treg cells. They have high levels of FOXP3, they're CD4 cells, et cetera. So again, our cells have migrated into the joint, they're there, and they still look like good regulatory T cells. And so we're relatively hopeful that as we go to higher doses, we'll see even more effects. And so that's, again, ongoing, and we'll have a lot more data in a year, but I just thought it would be useful to show how we actually really have now come full circle. Back in the 40s and 50s, when this peripheral tolerance was proposed and a Nobel Prize was awarded in 1960, you can follow this through, 65 years later, there's another Nobel Prize for being able to understand how this process now happens. And relatively quickly, at some level, we're now actually into clinical trials. And again, it's not just Sonoma. There's a number of groups trying to do this with their own little twists on this. And so I think in the next few years, we're going to have a very good idea of how well this works, where it works, when it works, and what we might need to do to make this even more efficient. And again, for me, this is very exciting because we're using this natural cell that has all the properties it needs, in theory, to control disease. And so what we're hoping for is not a treatment. We don't want to treat disease. We actually want to cure disease by resetting people's immune system back to the state it should be in and the state most of us have. So it's a goal, it's aspirational, but I don't think it's unachievable at this particular point in time. So I'm going to thank some people as well. Mary's already commented about David Gallus. David was a terrific scientist. He was a great mentor, a great friend. He's really, truly a Renaissance man at many levels. I actually have his art hanging in my house. I have a book of his poetry. David was truly one of the fine gentlemen around. Obviously, Mary's contributions, I don't think any of us would be standing up here if Mary hadn't been incredibly talented and done what she's done. Steve Ziegler, who's at the Benaroya Research Institute, was the first person really, I think, to recognize the value of the scurvy mouse. And he and I had long conversations about this. And he said, shouldn't we work on this? And I said, a little expletive, yeah. And Rolly Contri, who's somewhere in the audience, is really the first person to know that this gene was the thing that controlled regulatory T cells. And she'll never get enough credit for that, but it was tremendous work on Rolly's part and her team, which was fantastic. I'll also acknowledge the Sonoma team. Again, Jeff, who really got Sonoma going. Sonoma wouldn't exist. This whole process wouldn't exist without Jeff. Josh Bialke was the first employee at Sonoma, and he's driven so much of the work around this particular RA program and gotten it into the point where it could go into the clinic. Malin runs the clinical program, and Samir is the project manager. There's way too many clinical investigators to name, but watching clinical investigators go from cautious and not quite sure how they wanted to buy into this to now being full bore into this and wanting to treat patients and recruiting patients faster than we can make them is pretty impressive. And of course, Lars and Vivian here at the Karolinska, incredibly great collaborators. I will also make a little acknowledgement of two earlier mentors. Sid Golub was my PhD advisor at UCLA, really taught me the concept of perspective and being able to focus on something important. And BJ Folks at the NIH, who was just an amazing PhD advisor. I was her first postdoc, and I think we both learned how to work together as a team, and it was a great joy to work in BJ's lab. I will also thank the mice. No mice, none of this story. I and many others will correctly point out that mice are not a great model for human disease, but they really are a pretty good model for the fundamental tenets of how the immune system works. And we've learned enormous from them over the years, and I just want to thank them for that. I shouldn't say this with the mouse picture up, but I also want to thank my partner in crime, or partner in misdemeanors, I'm not quite sure what that is, which is my wife, Laura. She's put up with my inquisitiveness and my contrarian nature for 40 years, mostly with good cheer. And I wouldn't be here if she wasn't metaphorically standing next to me. And Thomas is my last slide. I mean, I'm over. But I was here in 2002. And I've mentioned a bunch of times when I was wrong, which is not an uncommon trait for a scientist. If scientists are always right, they're either kidding themselves or they're lying. So I'm wrong a lot. And I was here in 2002, and this is more my normal state. I've got sandals on. Usually I'd have short pants, but it must have been a cold day. But we were wandering around the old town, and we came across this travel trailer, which looks a lot like the travel trailer I was in when I didn't get the phone call from the Nobel Committee. But this one is from the Nobel Museum, and I stood in front of it and said, look, this is as close as I'm ever going to get. Apparently, I was wrong. Thank you all.
Dear guests, the 2025 Nobel Lectures in Physiology or Medicine has now come to an end. I would like to extend our gratitude to the Nobel laureates for their extensive presentations this afternoon. Thank you all for attending, and thank you for sharing this significant moment with us. When leaving the hall, please exit the same way you came in. Those of you continuing to the reception, please be prepared to present your ticket. The QR code will be scanned. And now, there is an opportunity to photograph the Nobel laureates on stage. May I ask the laureates onto the stage for a round of applause.